Hi everyone, here. Welcome to another podcast from We Talk Science. I am here today with Sue Ishak. Hi Sue, how are you doing?
Great. I'm really excited to be here.
Oh, excellent! I'm so excited to be talking to you today. Could you please introduce yourself to us, please?
Yeah, so I'm currently an assistant professor at the University of Maine in Orono, Maine, so we're kind of in the middle-ish. And as an assistant professor, where I've been for about 2 years, half of my time is spent teaching about animals and microbes to a variety of students, and then the other half of my time is actually researching those things.
Excellent. So, could you tell us about a little bit about your journey towards, where you are in Maine and how actually you got there in terms of your research as well?
Yeah, I'll give you the shortish version of that. There's been a lot of unpredictability. So originally, when I was a kid, I wanted to be a veterinarian. I wanted to help animals, and that was all I wanted to do. And I wouldn't take no for an answer. I was so sure that I wanted to be a veterinarian, and I got to the University of Vermont where I did my undergraduate, and I got a better idea of what it was actually like to be a veterinarian and what it actually meant. And I realized that I just had a completely fabricated understanding of what that was based on popular books and media.
Yeah.
Yeah, shocker, right? So I spent the next 4 years just taking different classes and wanting to work with animals and conservation in some way, but not really sure what I wanted to do. I specifically avoided anything with microbiology, genomics, or anything with sequencing or computer programming of any kind. I thought I definitely don't want to do those. And then, ironically, when I graduated, all I was qualified to do was to be a technician or an assistant at a veterinary hospital. So I did that for a few years until eventually I found a position back at the University of Vermont with my PhD advisor at the time, Andre Wright, who wanted me to do a project on microbiology, genomics, and computer sequencing. And I thought, well, I really want to go to graduate school. I want to work with animals again. How bad could it be? And it turned out to be the best decision that I've ever made. I really loved all of those things, and those have kind of been the center of my career since then. So my original project as a PhD student was to categorize the microbes that were living in the stomach of the moose. So moose, like cows and sheep and a bunch of other herbivore animals, but not all of them, have a really cool stomach with 4 chambers in it. And that first chamber is called the rumen, and it's like this microbial paradise. So it digests all the plant matter that the cow or the moose can't digest on its own, and it has all these cool functions, and all those microbes kind of end up acting like an organ and interacting with the animal cells itself. So it's a really cool environment to study. So I categorized what was in the moose. I grew some of those microbes. I made them into a probiotic and gave that to lambs to see if it would make them grow better. So I did all sorts of things, and when I graduated, my intention was to stay with animal microbes, and particularly with agricultural animals, because I particularly like that aspect. So I went to Montana State University for a postdoctoral position to do more data analysis on similar projects. So we would look at all the microbes in the rumen of various animals and understand how the effects of diet might change, which microbes were there, what they were doing, that sort of thing. As is somewhat common in science, I was there on a salary that was funded through federal funding or through grant money. And it ran out just sooner than expected, so I had to kind of scrounge around last minute and try to find other positions. And one of the collaborations that I had developed at the time, also at Montana State University, was working with soil microbes and understanding how wheat or different crops might select a soil community that they particularly like to hang out with, and how climate change might affect whether or not those plants can interact the same way with their microbes, grow the same way, be as nutrient dense, that sort of thing. So I spent another year at Montana State University looking at soil microbes. And then once again, I was there on grant money, and that started running out sooner than expected. So I found a position as a research faculty, which meant that I was a little bit more autonomous. at the University of Oregon in the Biology and the Built Environment Center. And so that was, by then, what, 2 or 3 left turns into my career. And I was looking at the microbes that you find in buildings and what humans are putting down and what we're picking back up and how microbes travel through buildings. Something that's been very relevant over the last year. Sure. But it seemed like very different from what I'd done before, but actually a lot of what you find in buildings is either coming off of you or your pets or plants or things that you're tracking in, or soil and water and air microbes that are finding their way indoors. So it actually kind of married the two, gut microbes and soil microbe stuff. So that was kind of cool. So I was there for almost 2 years. Third time's a charm, ran out of money sooner than expected. But it was really serendipitous because I happened to apply for, and eventually get offered this position at the University of Maine. So as I said, now I'm a assistant professor, which is like junior faculty, right? I've been here for 2 years. I've got 5 years to sort of prove myself, and then I essentially reapply for my job, which is called tenure. Yes. And if I'm awarded tenure, I get promoted and I get to stay. And if I'm not awarded tenure, I'll be finding another position. But I'm pretty confident that I got this.
Excellent. Oh, you got this. You got this.
Yeah.
So, I mean, listening to your history, basically, you're the common theme in all of the different aspects. You've been through agricultural animals. From starting from the moose to the agricultural animal to the soil to the plants and now to the built environment. I mean, the common theme in all of this is microbes and microbiology, right? So, what is the focus of your research right now?
Yeah, so I took one more left turn, so I'm actually back right where I started, back in animal microbiomes. Yeah, so I'm again working with livestock and also with some wild animals, and I've sort of expanded the species that I end up working with, the animal species. So I do still work on cows and sheep and that sort of thing, but I've also recently worked with some insects like fire ants. Mm-hmm. I've recently started working with lobsters and scallops that are involved in like aquaculture. So still agriculture, but with fewer legs. And so you're right. The common theme is microbes and how they're interacting with that animal, how they might be helping or hurting that animal, and also how they get to that animal in the first place. Because if you think about a cow, it's living at the farm, it's not running around in the woods most of the time, it's not going swimming. So there are certain microbes that it's likely to encounter in its daily life and some that it's not. Or if you're a lobster in a tank, the microbes that you acquire are from that water system or the facility that you find yourself in. So are we providing the right microbes to that cow or to that lobster to help them grow and be well and produce?
Okay, so your fundamental question essentially is how do you improve the health and I suppose the usefulness, I'm guessing, in terms of food source for animals that are domesticated, like cows and lobsters and other animals that we essentially grow for food. Is that correct?
Yeah, okay. And the usefulness is what kind of pays the bills. Right. But really it allows us to ask those broader questions like, what are microbes doing for an animal and why do we need them and why do we need certain ones at certain times and not others? And we can use animal systems because animals tend to live less complicated lives than humans, and we can use what we learned there to then apply it to ourselves and understand how we interact with microbes and how we might need them. So there's a surprising lot of information that you can find by studying lobsters or ants or cows.
Yeah. So that's really interesting because, I mean, whenever you look at like microbiome studies, right, people always look at mice. I mean, that is like, the model creature. And, we hope that whatever you see in mice is translatable into humans. But I actually was not aware that, there's quite a bit of similarities between, because, I mean, obviously cows, they have, cows and lobsters, I guess, both are, different. They live in a different environment. They eat different things. They are actually, grown, let's say, for different purposes than humans, I guess. So what are some of the similarities and differences that you actually see? Let's say, for example, if you look at a lobster in a tank, at your local Chinese store, food place or whatever, and how can you apply that to a human situation? Do you have an example of that?
Yeah. So this might be a bit of going out on a limb, right, to say that lobsters are like humans. But I think a nice way to look at that would be in terms of the built environment or those physical surroundings, right?
Right.
So a lobster in a tank can't really move to a different area. It can't control the microbes that it encounters. And even though those systems tend to be built to reduce disease and to control microbes, to a certain extent, you as a lobster do want microbes to come in, to colonize your shell or your digestive tract or to work with you or, whatever it is that they're going to do. So if you think about yourself as a human in whatever living situation you happen to be in, whether it's
Right.
a home or an apartment, city, rural, you might not have control over your local environment. You might be in very sterile conditions, you might be in very messy conditions, you might be in a degraded building where you don't have control over the microbes that are being put into your air and your dust. And those might have kind of health consequences for you that you're not really in control of. So there's a kind of a lot that we can think about in terms of how our environment, our environmental microbes might affect us by looking at
Right.
animal systems like that.
Okay, fantastic. Okay, so when you look at animal studies, animal microbiome studies, so for example with humans and things like that, a lot of people are looking at engineering the microbiome to create changes in the human sort of, physiology in the human animal. And I mean, at this moment in time, still, if I understand correctly, people are looking at the mechanism of how this might happen, but it's not always very successful, obviously, for various reasons. Has there been an increased sort of success rate in that sort of like modulation of the microbiome to increase, I don't know, in a cow and milk production, for example? Has there been any success stories there at all?
Yeah, so one of the, of course, big differences between cows and humans is that cows tend to live much more similar lives to each other than we do. So
Sure.
humans are immensely complicated. All of the things that we eat and all of the things that we do are very different. And even if we try to remember what we did in a day, we can't necessarily recall all of the little details that might
Sure.
have an effect on us. But, you tend to have groups of cows living under similar circumstances, eating very similar or the same diets. They might be more closely related than, say, a group of humans that you might study. So, the implementations that you use for cows or changing their diet or changing their living circumstances tends to have a more clear effect than it
Okay.
would do on humans. So, it's a lot easier to see what happens in a cow or a mouse or a lobster just because yeah, they
It's
don't go on
just,
vacations or
no,
have cheat
right.
days on their diet, right? Like,
Sure, sure.
yeah.
So that's like you were talking about the heterogeneity of the way that people live versus animals like cows, I suppose, or lobsters in a tank.
Yeah.
Oh, interesting. Okay, that's fair enough. Fair enough.
Yeah.
So tell us about the fire ant story. So I know that you've recently published a paper in the journal that I handle for work, iScience, about fire ants. Tell us more about that because I thought it was a really interesting story, but I want to hear it in your words.
Yeah, absolutely. So I sort of entered what was at least a 10-year story kind of at the tail end. So I will tell it in my chronological order and then
Okay.
backfill kind of from the beginning, right? So one of the classes that I teach is a DNA sequencing data analysis, which means that we've got strings of DNA code that are somewhat meaningless on their own, and we use computer programming and other sort of graphics programs to make sense of it all and understand who are there in terms of microbes and what they might be doing and whether certain things change it, right? So last spring, in the spring of 2020, I was teaching this DNA sequencing data analysis class as a draft version to the very first little cohort of students. And I asked around to some of my collaborators and some of my coworkers in case they had any old data sets that hadn't been published that they wouldn't mind donating. Because usually somebody's got some data lying around that they just haven't had time, and it just sits there for a few years. And then
Sure.
finally you're like, yeah, let me donate this. So one of my collaborators, Jean McCrae at the University of Maine, was kind enough to donate some of these ant samples that she had. And they turned out to be microbial communities from fire ants and then also from nematode worms that were infecting these ants, and then also from some waxworm larvae, which were part of some laboratory experiments. So I'll get to all of that in a second. So I gave this data set to two of the graduate students that were in that class, Alice Hotop and Sam Silverbrand. And over the semester, we worked through this. And at the end of the semester, they had drafted a manuscript. So we brought it back to Jean, and Jean said, "This looks great. This data actually isn't mine originally. Let me go talk to the original researchers."
Okay.
Yeah, who turned out to be Ellie Groden at the University of Maine, who's an entomologist who recently retired, and then also Patricia Stock at the University of Arizona, who's also animal microbiome entomologist type of thing. So Ellie and Patricia had been looking at fire ants for some time and talking about this and talking about nematodes for some time. And so they had a very different question than I initially understood. So fire ants are kind of misnamed. They're not actually properly fire ants, but they have that name because they've got a really obnoxious bite and they absolutely just sting you. They do tend to swarm a little bit. They will disrupt their little local ecosystem, so they'll kill off other ants. They'll disrupt how seeds are dispersed or which plants grow. So they can be obnoxious when they're outside of their home ecosystems. And fire ants have been introduced to the eastern coast of North America over the last 100 or so years, and they've come to coastal Maine, and they're just sort of a nuisance, right? Like nobody wants them here. And it was, who wants a stinking ant, really?
No.
So they've been kind of disrupting coastal Maine ecosystems for a while, and Ellie, the entomologist at the University of Maine, noticed that some of their colonies were collapsing. So it meant that all of the ants were dying off. And so she wanted to understand why, as a possible means of controlling their populations and getting them out of these areas where they don't belong. So she discovered that many of them were infected with this particular nematode worm that usually lives in the soil and then will invade ant tissues when they're juveniles or when they're young. The worms are young. They will hang out in the ant until the ant just dies of other causes. So it doesn't even kill the ant, it just waits. And then once the ant is dead, it just has a little party and grows and develops, eats that ant's carcass, and then emerges as an adult to go back to the soil and continue living on its life.
Wow.
And so the thought was that in these colony collapses where all the ants are dying off, it doesn't appear to be directly caused by the worm, but maybe the worm is actually introducing bacteria into that ant, and the bacteria is causing an infection that's actually killing the ants off. So Ellie had been doing some laboratory experiments for quite a few years based off of that, involving a couple of undergraduate at the time students. So Amy Michaud and Jonathan Dumont had done their undergraduate honors projects almost a decade ago now, looking at these ants in the labs and these nematodes in the lab and running experiments to determine, can these nematodes actually pick up bacteria from their normal environment? Yes, they can. Can they transfer them to waxworm larvae, which is like a moth larvae that's often used in the lab because they tend to be pretty well behaved in lab circumstances. They tried that. And then, of course, they sequenced the bacterial communities that they found in ants that they found from collapsed or collapsing colonies from the nematodes worms that came out of those ants, and then also from some of their lab experiments. So I got the full force of this backstory last summer, and in talking to the entire research team, we realized that quite a bit of this hadn't been published at all, and everyone was enthusiastic about moving this forward. So I spent last summer leading the charge to push all of these written pieces into one very massive manuscript and making sense out of over a decade worth of research. So it turned into this really cool story about how something seemingly innocuous might turn into be, some sort of biocontrol or some way to intentionally try to reduce these ant populations in Maine.
So, do you reckon that this has actually happened because these fire ants basically turned up in a place where they were not supposed to be at? So, this is not exactly their native area, if I understood correctly, right? Because I mean, this is you see this all the time where you have different species because of climate change, because of I don't know, humans traveling, between continents so often that there are species of microbes that are introduced in other places which cause can cause a bit of damage, for example, right? So, do you reckon that the reason why these nematodes are actually acting as a Trojan horse essentially is because the fire ants were not actually supposed to be there? So, you would reckon that they would probably not, be infecting other ants that are native to the region. And is that a correct assumption there?
Close. So I will
Okay.
remind our listeners that I'm not an entomologist. I usually work with warm-blooded things, so I might be a little fuzzy on the details. So the nematodes just kind of do their thing, and I think it's more that the soil bacteria from these regions is not something that these ants are used to seeing. So
Right.
they
Okay.
might be
Fair
getting
enough.
infected by other nematodes, but it's the bacteria that they're seeing that might be new. Because even though you might see similar types of these bacteria in soils from different locations, the very specific strain or the very specific environmental conditions that bacteria has grown up in will affect its capacity to infect things or different capabilities that it might have.
Fair enough. And it's just like the nematode is just acting as a vehicle to just deliver these unknown bacteria strains to the ants that basically cause them to die in the end.
Yeah, exactly.
Okay, fair enough. That's a really interesting story, as I said, and I think that the idea that that you have a population of another completely different species that moving into another area which is not native to them, can actually be controlled in different ways as well. So that's actually really really good to know. Okay, cool. So now changing directions a little bit. I would like to ask you how your experience has been as obviously a woman in science. With all the stories that people often hear. How has your time been in academia and in science? And following that question, what kind of advice would you give to a undergrad student who is a woman, or maybe even somebody from a minority group on how to continue in academia?
Yeah, so there's a lot to unpack there, and I would again remind everybody that my experience tends to kind of be different from everybody else's. So most of what I've faced hasn't actually been explicit sexism. Like, thankfully, no one has said anything weird or gross or inappropriate to my face, but I've certainly heard plenty of those stories. So in my experiences, most of that has been kind of on the implicit bias side. So implicit bias is more like acting on stereotypes and treating somebody differently based on who you inherently assume that they are. And a lot of this has been somewhat innocuous, or people don't even really notice that it's happening.
Yeah.
But I'll give you some examples. So there's been this assumption with many of the people that I've encountered, or people in general that sometimes work with female scientists, that we might be younger than we are, that we're a little bit less qualified, that we're not ambitious, we have less motivations. And so collectively, we end up being treated or thought of as less competent, less successful, with less leadership than we actually have. And this comes out in a variety of ways. So I've personally been paid less than some of my male colleagues in a couple of situations where my expertise was devalued over theirs. There's always a potential of being passed over for awards, which of course I wouldn't have heard about if I'm not getting the award. So, there's quite a few things that come up in terms of competing against your colleagues and this idea of merit. And if somebody doesn't inherently think that I have the same merit as a man, then they're going to rank me lower, right? And it very often comes out also in just subtle ways. So if I'm talking one-on-one with somebody, yes, I am a doctor, but they don't need to use my title. We can, talk as friends, as equals. It's totally fine. But the ways that people introduce a person to somebody else or to a group will set up the social cues for how that group should treat you. So in circumstances where someone hasn't introduced me with my title or with my job title or with any context whatsoever, And especially if that person knows me and treats me as a peer, then it sets that group dynamic up to also immediately treat me as a peer. And often that comes out as not thinking that I'm a professor. Often I've been mistaken as a graduate student at a variety of levels of my career, not thinking that I might be the expertise on that particular subject matter in the room, not thinking that I might be leading the project when I am. And so it ends up making my job harder to do just because of a simple slip of treating me slightly differently. And again, you don't even really think about it. But so for anybody listening, I'm telling you now, start thinking about it. Yeah, it's subtle, but the best times and the best job situations I've been in is where my gender hasn't even come up. Like, don't treat me like a woman. Treat me like I don't have a gender, because it honestly shouldn't really matter to my ability to do research or my ability to teach. It certainly comes up, and it can be an asset in many circumstances, but most of the time, don't even worry about it. Yeah. So my advice to people that want to be better allies or want to be better as scientists is to continue reading about this, continue talking to people, continue having these one-on-one conversations, and get other perspectives so that you have a better idea of how to be better. And my advice to really anybody that wants to get into science, but also specifically to those people that are from underrepresented or minoritized populations that feel like there's additional barriers, is to just make space for yourself and to make other people make space for you, right? Like, you have valid opinions, you have valid expertise, you belong in science, and they don't get to tell you that you don't belong there. So, speak up in meetings, ask questions, don't be afraid to contact people directly and ask for help, ask for advice, ask for guidance, because there are some wonderful people out there that will absolutely help you. And everyone in this industry has been helped by somebody else. I didn't get to where I am on my own. Many people helped me. So find somebody who's out there who's willing to help you.
That's absolutely great advice. I mean, I think that we are all aware, this is not just in academia, but also in every walk of life, that there are definitely disadvantages in terms of what you mentioned, in terms of salary, in terms of benefits and awards and whatever, that kind of thing, promotions, if you are part of the minority group, unfortunately. And I think that also as women, I think one of the things that we need to learn is, and tell me if this is something that resonates with you. We need to learn that we need to basically shout about our accomplishments, a lot more and a lot louder than we actually do.
Absolutely. I went into science thinking, and I guess that's probably how I approached most of my academic life, is that if I keep my head down, I work hard, I do a good job, and I'm smart, people will see this and they will recognize it and they will give me the awards. And that was probably true in middle school and high school. But once you get beyond that, everyone around me is incredibly smart and incredibly talented and does wonderful things. And so you end up not necessarily getting lost in a crowd, but you have to be your own advocate now. So you have to go out and tell people what it is that you're doing, and you have to market yourself, you have to advertise yourself, and you don't even need to be assertive when you do that. You don't have to be aggressive in any way, but you learn how to grab people's attention. So learning how to do science communications through setting up your own blog or social media sites can be a great way to help practice your voice with the general public, so that you then learn how to stand up to yourself when you need to have a conversation with the dean or something.
Sure. Wow. No, that's really, really good advice. I think that all of us could learn, how to do that. Maybe it should be sort of like a class or something, during undergrad or whatever, just to get people through this. I definitely I've had to learn that on my own. I'm still learning, to be honest with you.
Absolutely. It's been probably a 15-year journey for me to learn how to stand up for myself and to make space for myself. So don't feel like you will accomplish it in a day, but every day you get a little bit better.
Sure. Well, thank you so much. I'm going to say, I actually don't remember if I introduced you as Dr. Suzanne Ishak. So Dr. Sue Ishak, thank you so much for your time. I really appreciate it. Really enjoyed our chat. And, thank you so much for your time. The story that you're telling is so interesting. And I really, really appreciate, your advice for anybody who's starting out in science, or anybody really who, is in a minority group. I think it's very important that we get our voices heard.
Yeah, thank you so much for having me and giving me this opportunity to share my science and my perspective. I really appreciate the science communication that you're doing and also bringing up these issues because again, it can be hard to get a different perspective from your own, and so having these outlets like this can help share our stories.
Thank you so
Love
much.
it. Thank you.